Hello, everyone, and welcome to this presentation on flies in food facilities. I did take some liberties in expanding this topic just a little bit. Originally, it was titled Flies in Restaurants. However, part of my regular field activities is working in food processing facilities. So it's expanded a little bit more, but all of it will be applicable primarily to restaurants and food facilities in general. So from a standpoint of an overview, the things that we're planned to cover today, first off, we'll talk about why fly control is important. And I think in general, both on the part of our clients and the public, that there seems to be or can be a lack of understanding of why fly control is so important. And so we'll cover that. Then we'll go into identification, and my plan is to go over certainly the most common species, and we only have an hour, so we'll focus on the common species, but a couple of lookalikes or those that you may see maybe a little less frequently. We'll talk about how flies enter structures, because also, as I mentioned earlier, that there may be somewhat of a lack of general knowledge or understanding or thought about flies and their importance as public health pests. I think there's sometimes a lack of understanding as to how flies got into the structure, and then we'll wrap it up with control. So, significance of flies as pests, first and foremost, their importance as vectors of diseases. We know that the common housefly, for example, can transmit up to 100 different diseases, but our small flies as well. So not just those larger filth flies like the housefly that is easily recognized or more commonly recognized as a public health threat, the small flies as well can. So we know that even the fruit fly that's pictured there is capable of transmitting diseases, some of the foodborne pathogen diseases like E. coli and salmonella. So disease is important, especially when we're talking about our food facilities. Okay, so second up is myiasis. In many of these flies that we're going to talk about today will infest carcasses, so dead animals, right? Bottle blowflies, and we'll get into more detail on these flies, but bottle and blowflies, but even phorids. Phorids can be a problem in mausoleums, for example. So dead animal carcasses can be infested. Maggots can live in those dead tissues. On occasion, though, wounds, live tissues can become infested, or even if those eggs or maggots are ingested, sometimes we can get survival of maggots within the intestinal tract, and phorids would be an example there. So myiasis from a medical standpoint, physical contamination. So here we're talking about where we're getting insect or insect parts in food, infesting the food, or landing on our food, right? Getting bottled in product. And so we've got a picture there of mezcal on the bottom of the slide there. In that case, it's actually a larva of a moth, and it's accepted, right? People know that it's going to be there and should be there, but we can get physical contamination, flies, fly parts in food that are not intended and not accepted by the public. Increased spoilage. So many of these flies are going to be attracted. They're living in decaying organic matter. They're attracted to things like yeast and bacteria that are responsible for further decay of that food. And so they can pick up that yeast or the fungal spores or the bacterial pathogens and transfer it to food that's not in that same stage of decay, increasing and being more responsible for more rapid spoilage of things like produce. Brand and reputation. So just like we mentioned with the physical contamination, having those insect parts in the food. When we talk about restaurants, having flies land on a plate of food, just in general, does not give you warm fuzzies regarding what you might think about the sanitation and the condition of that facility. So it can affect our reputation, our feelings about that facility. I'd like to share a few commonalities when I'm going to break down. I'm going to talk about small flies and then the large, contrasting that with the larger filth flies. So talk about some commonalities of both, and starting with the small flies. So when I'm talking about small flies, we're talking about average size, 1/8 inch or less. That's kind of the dividing line. Moisture, we'll see this with a lot of flies, right? But moisture is going to be key in selection and survival of those flies in the areas where they're developing. Pupation, that pupal stage, there's 4 stages: egg, larva, pupa, adult. Pupation often occurs away from that developmental site. And that's important from the standpoint of survival. We see that with insects in general that have complete metamorphosis, that having that stage, that pupal stage away from the food, can help with survival, because if we eliminate that food source, we can still have a part of or a stage of that fly away from that food and still survival. Adults can be fairly long-lived. So with the small flies, some of them 30 to 40 days as adults. So again, even if we get rid of that food source, that source of the maggots, we can still have adult survival for a fair amount of time. And I don't know that everybody always recognizes that. Important point to help educate our customers regarding that, because even if they do the cleanup, right, we've gotten to the source, we found that rotten potato underneath the prep table, and we've eliminated that source, you can still have adults hanging around for a fair amount of time. And it doesn't mean that we didn't find the source. It could mean that we just have adult survival. I think sometimes with the small flies, we don't always recognize the fact that they are living. Many of them are living on the exterior of the structure. So I've got pear trees. I'm recording here from home. We've got a pear tree and an apple tree outside our yard. Nothing right now. It's December, right? But in the warm weather, we'll have pears on that tree, and we get bullface hornets excavating into the pears, and I'll see vinegar flies inside those pears. So some of these flies, even our red-eyed fruit flies or vinegar flies, can be outside and enter from the exterior. When we have small numbers, we're looking at our insect light traps, let's say inside, we see 1 or 2 phorids or 1 or 2 fruit flies. It may mean that they've just come in from the inside, that we may not have a source. We need to be looking for larger numbers. And they can build up in large numbers in short periods of time. That's why we have these large problems with our small flies, typically more in the summer, right? They've had that opportunity, those ideal temperatures. The female is laying, let's say for fruit flies, 500 eggs, short developmental times in that, let's say 8 to 10 day range. And by the end of the summer, or even mid-August up here in Chicago, we can see large numbers as those generations turn around and those large numbers of eggs and maggots develop to adults. All right, so then contrasting that with our large filth flies. So here we're talking average 1/8 of an inch or more. Although we'll have often with the small flies populations, I mentioned they could come in from the outside, right? We often have developmental sites also inside, greater potential for that with the small flies. However, with the large filth flies, and in particular, when we're talking restaurants, very rare to have house flies or bottle flies on a regular frequency developing inside those sites. Not that I haven't seen it, right? It can happen, but more typically they're developing outside and coming inside, and exclusion is more important with the larger fill flies because of that reason. They too, like the small flies, moisture is going to be key in developing. We'll see garbage, carcass, and manure. And as I go through the different flies, they'll be fairly common that they can develop in all three of those sources. Pupation, just like the small flies, can occur away and will occur away from the developmental site. It's a really moist developmental site. They're not going to, from a survival standpoint, do very well as a pupal stage in that moist environment. It's to their benefit to move away and pupate away from that moisture. And they will build up in large numbers, just like our small flies, in short period of time, rather short turnaround time in life cycle, developmental time, and a lot of eggs, kind of in that 500 number range being laid per female adult. All right. So identification, small flies, and I'm breaking it down into three main groups. The most common are fruit flies. So this is the Drosophila flies, our phorids, and then our drain flies. Just like the fruit flies, the Drosophila flies, this is not just one species, there's multiple species. When we say phorids, it's a family of flies. It's not just one type of phorid out there. And then our drain flies, the same thing. And then I'll go over our lookalikes, our fungus gnats, and I'm going to cover small dung flies. So as I mentioned already, multiple species of fruit flies, and something in the range of about 170, 175 different species of Drosophila flies in North America. But we're going to focus on these as most common. So Drosophila melanogaster, the fruit fly, we often call it vinegar fly. A lot of times I'll refer to it as red-eyed fruit fly to distinguish it from the dark-eyed fruit flies that I have listed there on the slide as well. So our dark-eyed fruit fly, and then its lookalike is the Drosophila hydei. May not be as critical to know if it's dark-eyed fruit fly or Drosophila hydei, but know that in general, our bright-eyed red-eyed or bright red-eyed fruit flies versus our dark red-eyed fruit flies have a little bit different preference in the type of food that they're going to lay their eggs. All right, so identification. On the left-hand side of the screen, we have the red-eyed fruit fly, tan in color, this striped appearance on the abdomen, differing from, on the other side of the screen, the dark-eyed fruit fly. It may look almost black when you look at it from a distance, right? The eye, but it really does have some red pigment to it, and it's kind of I would describe it kind of as a maroon color. It also has this, instead of that solid tan of the red-eyed fruit fly, it has this kind of mottled appearance, kind of a marbled gray black appearance on the thorax behind the head. And those are the keys to identifying it. I also put in a slide showing hydei, very similar again, types of food and food preference for it to be developing in. But on the left-hand side, you see the darker coloration of the front pair of legs versus hydei, slightly lighter color, if you want to go ahead and ID those separately. Next to the adults, the other common stage that we often see, first adults, right, more common than the pupal stage. And again, I think that has to do with the fact that it's going to migrate out of the food source you can dig into that gunk or food that it's developing in and find the maggots, but it's a little easier and sometimes a little more evident to see that pupal stage. Looks kind of like a seed. You might describe it as a seed. On the right-hand side, it's showing the life cycle of that fly with the adults and then the maggots. And then you see that pupal stage, that reddish brown color with these two kind of horns on it. And those are characteristic and can be used in identifying the pupal stage. A little bit on the biology and behavior differences between our red-eyed fruit flies and our dark-eyed fruit flies. So a little shorter life cycle on the red eye. We're talking in the 8 to 10 day range. I describe their behavior, and again, this can be helpful in identification, is a little bit more skittish. If I've got some peaches on my counter that might have some red-eyed fruit flies and I approach that area, they tend to fly off. And we see with dark-eyed fruit fly, not so much. They're not that skittish. They'll often rest on wall surfaces close to where they're harboring. You can almost go up and touch them before they'll move. So that's a behavioral characteristic that above and beyond what they appear, their appearance can be used. I should mention on dark-eyed, average length of life cycle, 21 days. We said 8 to 10 on red-eyed. As far as food preferences, red-eye tends to be food in the early stages of decay. So we're seeing it in that overripe fruit, right? The peaches that have started to overripen, a banana. We'll see it in sugar that's in the fermenting stage. So you may have heard of sugar snakes in pop machines, the drains, that accumulations of sugar where it starts to ferment. One of the reasons that we have problems with this fly in bars is because of those fermenting liquids and the attraction there. They're feeding on the yeast in that food. We recognize it as human food. Dark-eyed, not that they can't be in some of the foods that are not completely decayed, but tend to be more greater stage of decay, the food preferences for this particular fly. So food sources that I might describe as gunk, things that we wouldn't necessarily recognize as human food. One of the reasons that we see dark-eyed fruit flies more in floor drains and deteriorating floors is because of that. And if you think about that, you're not getting a banana in a drain. Hopefully not. It's food in a greater stage of decay. Okay, so phorids. Again, this is multiple species. There are two common species that we see mostly in structures, but there's more than one phorid possible. In the video that you're seeing to the right, just like I talked about some of the behaviors as useful in identification. Phorid flies are called scuttle flies as well as humpback flies, humpback referring to their body shape, but scuttle, that erratic behavior that you're seeing in the video. And sometimes from a distance, I can see that behavior, not even having picked up the fly to look at it, but just that erratic behavior is a key to that it's phorid flies. On the picture to the left, showing a phorid fly close up, couple of characteristics. That hind pair of legs and that first segment coming off of the thorax is the femur. And much like you would expect a grasshopper or cricket to have that enlarged first segment on the leg, phorids have an enlarged femur. So think of a flea or a grasshopper, very similar. So that's one characteristic to look at. The other, and the most common, I would say, characteristic that we would use is the wing itself. And we've got a picture, a close-up of the wing, the bottom of the screen there, that enlarged veins at the very top margin and no cross veins on the wing, so no linking between the veins, but that enlarge or that uh, darkening of those top veins, very easy to see on that fly. And again, like our fruit fly does have those horns on the pupa, and got a close up there. Our last of our common flies, drain flies, pictured here. I sometimes describe those wings as kind of, or the body shape is like a stealth bomber, the way they extend to the back, but some people will describe this as kind of a heart-shaped body shape to them. Also very fuzzy. Wings, the veins on the wings of the drain flies, or sometimes called moth flies, are going to be lined in parallel to each other. This behaviorally is a very sluggish fly, and they'll land on wings. We talked about dark-eyed fruit flies, or dark-eyed fruit flies landing on walls, right? The same will happen with drain flies. You'll see them congregating on walls, and you can go up to them and slap on them and kill them. Okay, they're not even flying off. They're a very sluggish fly, and they'll leave kind of a dusky residue on the wall, and that comes from those hairs and the scales on the body, called moth flies, because they look somewhat moth-like, but they are a fly. Drain flies are living in biofilm, and I call it the slime layer, typically, that you'll see in a drain. So it's a film that's caused by bacteria. And I think from a public health standpoint and disease standpoint, it's important to point that out to the customer, not just that they need to remove that, but that they have pathogens. They've got slime and a buildup of potential foodborne illness pathogens, including things, again, like salmonella, that's incorporated into that biofilm. Needs to be cleaned, not only because it's supporting our drain flies, but it's also supporting pathogens. Larva, and talked about pupa being maybe more common as that second stage beyond adults to be seen for the phorids and our fruit flies. But with drain flies, I think it's larva, and very, very characteristic, because the maggots of our phorids and our fruit flies are going to be light in color. These are almost black and fairly large, and never fails that I'll get someone of our technicians saying, we've got these black maggots or worms in a restroom, around a urinal or something like that. And without even seeing a picture, of course, we got to confirm it, but 9 times out of 10, it's drain fly larva, because of that dark color. Is very, very characteristic. All right, small dung flies, sometimes called sphaerocerids, because, and why people will use that term rather than small dung flies, there's other flies that are called dung flies, right? So it helps differentiate it and be more specific. About 1/8 of an inch in length, very, very common, right? We're talking about all of these small flies being 1/8 of an inch or less. Black to dark brown in color. The antenna relatively short. I should have pointed out on our drain fly is one of the smaller flies that have long antenna, but small dung flies, small antenna. Decaying plant material. These can be carcass feeders, can be associated with manure and feces as well. Blocked and damaged drains. So this can be one of those flies. Phorids, the other one that sometimes we have problems where there's been a pipe break. We can have sewage or other food leaking into a crawl space or subslab, and these flies breeding in large numbers. So small dung flies and phorids there. Can be a pest of poultry houses, hog barns, etc. Key to identifying small dung flies, besides the things I've already mentioned, is the legs. And in particular, that last pair of legs, it's got an arrow showing it, and then it's circled in the picture below how that leg is configured. The last few segments on that leg are tarsi. The first two coming off of the leg there are kind of enlarged. Okay, so that's what we're going to look for and key in on when we're trying to identify our sphericerids or small dung flies. Last of our small flies that we're going to talk about, dark-winged fungus gnat, often associated when we say fungus gnats, house plants, maybe atriums, but they are pests of greenhouses. So if you do any facilities that are growing lettuce under glass or tomatoes, you can see fungus gnats associated with that. And then roofs with leaks. And in particular, when we've got these flat membrane roofs, and we will see this in food processing facilities, pharmaceutical facilities, packaging facilities, where those roofs over time develop gaps, leaks, and we get fungus gnats developing in and underneath that membrane. Fungus gnats are really very, very common, okay? They're light attracted, and when I look at insect light traps, very often can see fungus gnats in those light traps. So they're coming in from the exterior, fairly common, but when we're seeing large numbers, we've got to look for sources internal. In restaurants, it could be planters, right? If there's flat roofs, that's another area to look. And then on the exterior, mulch, areas where organic material might have accumulated. Moving into our larger flies. So here we're going to talk about flies that are over 1/8 of an inch. We'll talk about house flies, 2 filth flies. There's more than just 2 types of filth flies, or flies that are categorized as filth flies, but we're just going to focus in on house fly and bottle fly. And then I'm just going to talk a little bit about the overwintering flies, the cluster flies. So house fly, little over 1/8 of an inch in length, has these 2 dark stripes on the area, 4 dark stripes on the area behind the head. And they'll have on the abdomen quite often a yellow patterning right underneath the wings that is visible underneath the wings. Pupa, again, could be the next most common stage that we see. Not that we wouldn't ever see maggots, right? We can see maggots if we get down into the breeding area, but the pupa, fairly common, and can sometimes be confused with othecas of, let's say, American cockroach, but you're going to see those striations or lines within that pupal case, as well as at the end there, and pointed out in that picture, the spiracles that show up kind of look like little eyes. Our blow and bottle flies, color is going to be the main key. Certainly there's wing, if you wanted to look at wing venation to confirm, and if there's any question as whether or not it's a blow or bottle fly, that can be used. But color typically makes this fly fairly easy to identify. It's got that iridescent or metallic coloration, can be green, can be blue, can be a kind of a brassy color, or even there's a black blow fly, so a black kind of iridescence to it. Primarily carcass feeders, although again, garbage and manure, just like our housefly, common sites for development. And then our cluster flies. So actually cluster flies are in the same family as our blow and bottle flies. And I want to point out here that there isn't just one species of cluster fly, although we seem to think that there is sometimes. There's multiple species, and some of those species, what it's developing on is not well understood. There's some indication that yes, earthworms right, can be a source for the main species, Pliniorudis, but there are other species of cluster flies, and bees have been implicated as a possible source of food that they're parasitizing bees, and then caterpillars the other. So know that there's more than one type of cluster fly, okay, that could be infesting structures. And I'll put a link to a key to cluster flies that also has, along with identification, some geographic ranges for the different species of cluster flies. Not well studied, opportunity there to better understand what species there are in the US and the food sources. Okay, as far as fly management program, we're going to start with inspection and assessment to determine where the flies are potential sources. If they're coming from the exterior, what those avenues might be for fly entry. Then once we have determined where our sources might be, where the possible routes of entry are, determine and implement our controls, come up with what our control plans will be, and then implement those. I've highlighted monitor there as our third step, and I think that's an area as an industry that we can improve upon. And so I'm going to talk a lot about monitoring devices, and then additional action. So our monitoring devices are going to tell us what additional actions might be necessary based on our monitoring results if we're seeing reintroduction or measuring less than effective control through the use of our monitors. Additional actions are taken. So those are the 4 basic steps. Why monitoring is important? Well, number one, it can aid in identification by trapping that insect. Not always do we go and the insects may be easily found, right? So having monitors can help us determine what pest it is. It can then help measure control results. Have those programs that we've designed, are they being effective? Aid in pointing to the source. So use of multiple monitors can help us pinpoint where that source is. If we have some difficulty or challenges in finding that source, we can again take additional actions based on what our monitors are telling us, and they can provide data over time. So did results improve in 2022 over 2021, for example? Are we making progress? Is there continuous improvement in our control programs? Light traps is a primary monitoring tool that we can use for flies, and they do a really good job on forage. Forage highly attracted. You can see hundreds and hundreds of forage flies in a light trap. Fungus gnats also very highly attracted to light traps. Small dung flies, drain flies will go to light traps. Larger filth flies, or a house fly, or bottle flies, or cluster flies. But fruit flies, not so much. There's a small window of time right after they emerge from the pupal stage that they are light attracted. So typically, we're not seeing hundreds of fruit flies in a light trap. And that's a problem because light traps are very effective monitoring tool for flies, but not so much for our fruit flies. So what can we do about fruit flies? And unfortunately, I'm going to say there's not a really good alternative. We can use sticky traps, and there are some commercial lures available that can be used in conjunction with our sticky traps. So we can take advantage of that. They tend to have to be fairly close, though, to where that source is. So you might have to use a lot of them. Jar traps, and to the right there, we've got that apple trap with the liquid. Yes, we can trap them with those jar traps, but they tend to be messy. tend to be difficult to count the number of flies in there, right? It's not what I would call the ideal tool. And so it'd be nice, glue trap good, if we can get them with a really good lure. We can use light traps, but understand their limitations and that short window of time that the adults are going to be attracted. So better monitors needed, if at all possible. And hopefully the manufacturers can address that in the future. Sticky boards, there are some flies that will be attracted to sticky boards and fungus gnats, both in kind of the fluorescent green color or bright yellow, can be highly attractive to fungus gnats. And so they're used in greenhouses, but can be useful. Again, you can use light traps, but colored boards, also very effective for fungus gnat. Houseflies, sometimes sticky boards, think about fly paper, not just boards, but fly paper can be an effective tool for monitoring houseflies as well. A technique that's used more in agriculture is counting of fecal spots, fecal and regurgitation spots. So again, not so much in our structural field and not so much in restaurants or food facilities, but in ag, let's say a poultry house might be a viable tool, where a white board, because they will both defecate where they land and regurgitate. You can count spots over time, how they increase as a measurement of a success in fly control, or as a threshold for trigger for treatment. More of a what I would say an inspection technique versus monitoring, but I did put this in here. The use of either clear plastic tape, saran wrap. Sometimes people will use a clear cup over a drain to see if that area is supporting flies, if flies are coming out of, let's say, a floor drain. For actual inspection, variety of tools. So a good flashlight to start with, right? I've got a picture there of a very strong flashlight, a hand lens, and then I put in parentheses digital scope if you can have one of those available. But a hand lens, to me, and I've highlighted that because hand lens is second to flashlight as the most important tools, especially when we're dealing with these small flies. You can tell a bottle fly right in a light trap, but if you need to identify fungus gnat and confirm it, forward flies versus other flies, small dung flies, we've got to look at those tarsal segments, right? You're not going to see that with the naked eye. You need to have a good hand lens, relatively inexpensive. You can buy them on Amazon for under $10, and even a light incorporated in with that hand lens. Gloves, we may be examining and looking into areas that, I'm going to have to cough, sorry. I apologize. Areas like floor drains where there's pathogens, right? So you need to protect yourself through the use of disposable gloves, multi-tool. You're gonna have to get into things, right? Scraper, spatula, getting into that gunk, maybe putting on an index card, the next item I have there, seeing if you can find maggots. Knee pads, because some of these areas are going to be lower, and to help protect your knees. Fiber optic scope, more of a I would say a less common tool, but sometimes needed if you want to scope into an area to get a better look at, let's say, into a drain system. Sanitizing wipes. You're going to use that scraper to go into a drain. You won't want to clean it off, right? And make sure that you're not transferring those pathogens into the next item or next area that you use that scraper or spatula on. Again, we've got foodborne pathogens. And then vials for collecting samples should you need to have somebody take a look and confirm that the ID. As far as food and stage of decay, and I've put this on more relating here to our small flies, Although I've seen some papers that say phorids will attack a totally intact and um, not overripened banana, for the most part, we see that there is a commonality of decay and level of decay in which that small fly will infest and develop in. So our small fruit fly, early stages of decay, again, looks like food, okay? Dark-eyed, we're getting more into a further stage of decay. Phorids, primarily a greater stage of decay, kind of similar with the dark-eyed fruit flies. And then our small dung flies, and we're talking about manure and a larger level of decay of that food. Dispersal ranges, and again, there can be some variance in this, right? I've seen some literature that said red-eyed fruit fly is as far as 6 miles can go. But for the most part, and based on practical experience, when we're looking at structures, those red-eyed fruit flies fairly close to the food source where they're developing, and the same for drain flies. Not far from typically are those drain flies moving. You can get drain flies coming in from the outside from a distance, right? Red-eyed fruit fly, but when we're talking about structures, typically fairly close. Dark-eyed fruit fly maybe a little further. Phorids, we had a hospital where we had a phorid fly infestation traced to a grease trap in the basement of this hospital, yet we were getting phorids on the second floor operating room, quite a distance. But that was the source. So I'm saying phorids maybe a little further as far as distribution, and we know our large fill flies, excellent flyers, can be coming miles, right? And phorids as well. Documentation that phorids will fly miles. So I'm putting that at the furthest from the source as far as dispersal range. When we're doing that search for the source, I just wanted to mention that take into account attractant odors, right? So there may be some pull away from that source. Both dark-eyed and red-eyed fruit fly attracted to fermenting beverages. I have a son that actually, Ben Hoddle, that provided that video that did his master's on attractants for dark-eyed fruit fly, and red wine came in as the #1 food attractant. But my point here is that some of these food odors can also impact where you're finding the flies, and it may or may not be the source of the flies. So keep that in mind. Fly specks, so they're going to poop. We showed a fly card, right, that could be used for counting, for monitoring. Fly specks can also be used as a cue as to where the flies are landing and potential sources for them. Moving into paths, or how they're getting into structures, or how that infestation, that problem may have originated. So we can, in the case of restaurants, for example, or grocery stores, supermarkets, where we're getting produce in for the smaller flies, we can get introductions in that way. We can get introductions with returned product, and for example, some states have recycling where consumers are paid 5 cents or whatever it be to return a container. Okay. I was just speaking in New York not too long ago, and they still have that system in place where you return your recyclables, and they see small flies as well as German cockroaches associated with that. From a standpoint of paths into structures on the exterior, so of course, open doors. Our food facilities are in business, right? So there's going to be some opening of doors, right, for people to come in restaurants or for deliveries, but keeping doors as closed as much as possible, closed when not in use, because they can come in from the exterior during the warm season. Lights and fungus gnats are more commonly or primarily dusk flyers. So we can get some impact of the wrong type of lighting being used on the outside of the structure and attracting those insects towards the building. We want to use sodium vapor versus mercury vapor lighting when possible. Mercury M, more attractive than sodium vapor lighting. Many of our clients now using LEDs and looking at the type of LED, trying to match up that spectrum of light to match a less attractive LED. Important there. We have manufacturers that are developing LED lights for light traps, right? So we know that LED can be attractive, typically less so, but we still want to try to do our best in matching up the light that is less attractive to insects. If we can have lights that shine on poles or mounted on poles that are shining onto the structure versus mounted on the structure, better. That picture on the bottom there is a dumpster, okay, and lots of what I'm calling gunk there. We can have not only dumpsters attracting insects like our larger fill flies, but providing developmental sites. Those pads under those dumpster compactors often have cracks, often don't get clean. We can get accumulation of organic material, and during the warm months, if you're there when the dumpster box gets moved out, you can find maggots. Okay, so we've got development right outside, and typically right next to that dumpster compactor is going to be a dock door. Okay, so look for that. The importance of that is an attractant. Flies, fill flies, house flies, bottle flies out in the environment, smelling those odors if it's not kept as clean as possible, and then laying their eggs if that food is not getting cleaned up when that dumpster gets moved. I also think there's some potential that if the dumpster box itself gets just dumped but not clean, that we can have flies. Immature stages, pupa, inside that dumpster box, and food residue. If it comes, and I've seen this at a packaging facility that we do, that a dumpster box that's showing a food processor name on the outside showing up at that facility. Well, are we moving flies in that case? If that box didn't get cleaned prior to arrival and being brought in by the waste hauler, could we be bringing flies with it? So look at that as far as reducing flies. Plumbing, septic systems. So many of these flies can come in through plumbing. We talked about the potential for breaks in plumbing and how that can impact our fly issues if they're not keeping the drains as clean as they need to. If the water, the P-trap, the water level is not maintained in the P-trap of the drain, or that drain was not designed with a P-trap, and so it's just a straight pipe, we can get flies coming up into the structure there. Talked about dumpsters already and the importance of cleaning. I tell our techs, if at all possible, try to show up, schedule service if you can, as much as possible when the waste hauler shows up. It is amazing to see what a dumpster pad can look like and the importance of that relative to the fly pressures inside the structure. Talk a little bit about some key areas. Talk about cleaning, waste, damage or infested product, food service, and food prep. Important to note, not all cleaning strategies are going to be good, okay? You need to sometimes provide very thorough instructions to your client regarding how to clean. They may not have that understanding of what we might say fly clean. And I've got a picture there of as an example, an illustration. Okay, so leaving your dog at home that might not be, let's say, housebroken, with the Roomba scheduled for starting up while you're not there, can result in some problems. And so we've got a Roomba spreading fecal material. And search it on YouTube. There's lots of videos I didn't include here. So other examples of the Roomba isn't necessarily something that relates to our food facilities, but this does. Water under pressure. So sometimes the cleaning strategy is the use of water under pressure, and clients need to understand how that use can impact potential infestation. So we could be pushing organic material as well as moisture to areas that don't dry out, moisture key for development, or that we don't see or clean. So in this picture, that pipe on the floor, if we're getting water, organic material that's sifting under there, less likely to dry, remain moist, provide breeding sites, hard to clean. So raising up in that case, that piping off the floor would help. Other areas more relatable to restaurant, for example, behind equipment, those ramps to reach in coolers or walk in coolers, typically not well sealed. We can get moisture material under there. The other problem with water under pressure, we can get cracks in grouting, can add and contribute to floor deterioration. We've got some pictures there. We've got a floor tile that's been churned over, fly pupa there. Important to note that those tiles are typically glazed. When that glaze comes off, we can get some moisture getting down in there. Remedies, of course, structural repairs, sealing those areas if they have become open. Great if they can put equipment on wheels so it pulls out, so you can help with both cleaning and drying of floors, and then supplementing with fans. And the picture to the left is showing one such, what we might call a squirrel cage fan, that can be directed to areas where we're having hard time after mopping is done, that a fan can be used to help dry. Some sanitation fails, and this may relate a little less to restaurants, but I see this out in the field where maybe a warehouse is built. They didn't plan on using floor scrubbers to start with. They bring in floor scrubbers, but they've got no way to clean them, no way to properly, or the right location to empty them in. And so we get areas where there's water accumulating, we get organic material. Picture to the left is a 55-gallon drum where this was that situation in that facility. They have a good place to dump the contents of a floor scrubber. They're putting it in a 55-gallon trash container. The problem is they weren't emptying it, and I was there on a mouse problem. I just kicked the can, so to speak, and flies blooming up. Got to have a better system. We also know, and this does relate to restaurants, mop sink areas, that's the picture to the far right, tend to be areas where there are a lot of materials, hard to sometimes see into them. Water, moisture, structural components can deteriorate there. The baseboard has come loose, and we've got small flies in that area. Dumpsters, this is to the left. One of our supermarkets was actually composting a vegetable material. We got a little green river, like we like them in Chicago for St. Patrick's Day. This green river underneath that dumpster, lots of small flies. Bad plan there to be composting without a plan to clean it. Some of the bigger carts, and that's the center picture. Again, the same thing. Sometimes they start using these larger rolling carts, right? Didn't anticipate initially to be having those on site, and may not have a good way of cleaning those. And if we get some residue, we can get food residue that stays moist. We can get small flies. And then that's a trash chute on the right. That's an actual hopper that lifts up and dumps the food for repurposing, recycling. Good plan to repurpose food, but very, very difficult to clean the sides of that, and we can get small fly problems. Similarly, trash chutes, and looking for not only internal and how you're going to clean those trash chutes, but dumpsters that connect to those trash chutes and any openings where we might get flies. Again, those areas prime for fly attraction, and then openings permit fly entry. Here we have grease traps, and I mentioned that hospital. We do see forward fly associations. We've had grease traps leak, subslab fly associations in the subslab area. And then that's a wastewater treatment area and a food processing, trying to take out some of the solids before it goes into the municipal sewer system, but that needs to be cleaned, right, to prevent flies. Drains are complicated, lots and lots of gaps. I've talked to food processing facilities and facility managements for restaurants and said, can you redesign this? There's too many nooks and crannies for areas to build up that are hard to clean. And unfortunately, that's the way it is. Sometimes they can't retrofit it, but there's lots of areas where we can get leakage and small fly development. Channel drains are the worst. I got some pictures there of channel drains. Those are the long drains, right? Tend not to drain very well. We can get accumulations. That center picture is showing an area of floor deterioration. There's lots of floor drain contact area, right? That needs to be maintained, and unfortunately not always done so. One-way seals, liquid breaker green drain valves can be used so it allows the water to go down, but flies not coming up. I wouldn't use them in an area of a lot of solids because it's a fairly small membrane that's the valve, and it could stick open. But great in restrooms, for example, where you don't have a lot of water flow and you may not be maintaining that water gap in the P trap, right? This can help solve that. And then bioremediation. Recyclables and repurposing it, I'm getting down to the end of my time, but sometimes not always handled as well as they should be from the standpoint of it's considered waste, right? So they're not as concerned about it, but we need to make sure short life cycle of our flies, the small flies in particular here, we're talking about with the recyclables, that we're making sure that we're keeping that in mind and cleaning out and emptying those recyclable containers. That trailer is showing sometimes we'll have facilities that have large quantities of food, and leaving the trailer doors open for that loading and unloading over time, we can get flies coming in. Damaged product. Pallets and recoup, how that's handled, right? Some of our warehouses, if you've got a damaged container and subject to forklift and rough handling, right? We can get the picture to the left is forward flies, damaged containers. Sometimes it's a packaging seal issue though as well. We've seen this with kind of the juice box type containers where you might have a seal break. It can happen with cans as well, but you get a break in a can, and maybe in the middle of that pallet, very, very difficult to detect and find that. But we've had situations like that. Food service and prep. I've tried to research how to clean a drain, the drains for both the pop and the beer taps, right? You can't find it. And the fact that I can't find it with a search lets me know that probably people are not doing it. So we can see for small flies here, we're talking about associated with beer taps, pop or soda taps as well. Picture to the right is a food service counter, getting back into our restaurants. And sometimes the seal there can be imperfect. If you can get your head up or use an inspection mirror underneath, sometimes you'll even see some seepage, right? As that counter gets wet, if there's a accumulation of organic material and that moisture stays in there, we can get small flies. Insecticidal adults, ULV, fly baits, resting site treatments. And by resting site treatments, I mean, we know that the flies are resting in a certain area. Treat that with a sponge. Some people call these wipe downs of that surface, and we're just getting a kill of adults by doing that, right? But I also said that sometimes our adults are hanging around long past when we got control of the source. Ultimately, getting source is what we want, right? We want to get to how those and where those flies are coming from and control it at the source whenever possible, especially when we're talking small flies. Can be difficult with our larger fill flies. IGRs and fairly new are some of these dual products, right? So it's more than just one IGR perhaps in the product, or an IGR with an adulticide. Take advantage of those. Residual insecticides and sanitizers. Future LED traps, okay? And we've done some work and started to work on effectiveness and measuring effectiveness of these LED traps to different species. At least with the filth flies, we're seeing comparable results, and that's a good thing. At least one manufacturer is working on a repellent. So an adulticide repellent with an IGR is a possible solution for drains. So let's say we've identified a floor drain as the source, using that repellent to keep the adults away and adding that IGR to kill the immature stages. And last point, upside down IPM. I know, and it is the right strategy to target source reduction, right? Getting to the source when we're talking our small flies, getting to the source with our larger fill flies, and the importance of exclusion as part of our program. Those are the foundational elements, right? However, sometimes we have to get quicker control. Some of the structural repairs or other controls that might be needed can be quite expensive. I talked about fungus gnats and roof. That's a major capital expense for a customer. We may have to use the upside down IPM approach, where pesticides are used to start to get relief and get as much control as possible as we wait for that ability to make those longer term controls. Subslab fly problems, the same thing. That's a capital expense. It's not going to happen the next day. And sometimes we also have health department or regulatory concerns that we have to address through the use of pesticides, where a health inspector says, I'm going to come back if I see one fly, I'm going to shut you down. Those kinds of things. Pesticides may be the first item used as we still work on the importance of source reduction, the importance of exclusion, but starting with pesticide to get relief. So sometimes it's an upside down model. All right, summary. Sanitation efforts can both help and hinder control. Got to use the right sanitation techniques, right? Fly maggots need moisture. So when we're looking for flies, we're looking for those moist sources. And moisture reduction, drying out an area, can be part of control. Insecticides can and may be required in control, and identification of species is critical for our control plans and for tracking, trending, and monitoring, right? All right, and I have one reference here. However, I will put additional links into Whova, so look for those. This link, Penn State has done a remarkable effort and placing a bunch of videos for different types of filth fly control, the larger filth flies, but in different types of facilities. So I encourage you to go there, and kudos to Penn State on that, but I will post some additional references.